So first of all, welcome, and thank you in particular to Judy Beck, who's at the back of the room, Brian Cummings, who helps to set all of these lectures up and is in charge of our communications, and to Kyle Good, who's at the back of the room, because without UCI Media, as you can see, we cannot handle these technical details. It's very critical. One of the reasons I wanted to talk about Craig and the things that he's done for the center, and I'm going to just highlight those couple of things and not give my sometimes big, long introduction about science and why it's important and why academic research is important. But one of the ways that all of those things are important is that we have to leverage funds for everything that we do. So we were very fortunate to receive an endowment gift from the Cullens 2 years ago, and we turned around and invested that in a new instrument in our core facility for mass cytometry. That was great. Craig was incredibly important, along with a number of our faculty on our shared research labs team, to be able to get that off the ground. But he also took on heading up an application to the National Institutes of Health called an S10 large equipment grant, and with that, we were able to secure an even better gizmo that links to the first one called the Hyperion, which is a way to be able to look at proteins, 50 proteins simultaneously with spatial distribution in a piece of tissue. And that's launched a new core facility for us, which he is working with quite a lot, as is Matt Inlay, who's back in the room there. Give wave, Matt. And that new core facility has now been used by at least 16 projects across the whole campus. So just one vignette about why those investments are so incredibly important to us, and not just us in this building, but really science writ large across the campus. And so I wanted to highlight that. Also really important for us, with the refunding of CIRM at the end of 2020, we've spent a lot of time over the last year, 18 months, gearing up to be able to take and maximize the advantage that we can take from that funding. I think I've talked before about the training grant that we received last year, which was the top scored in the state, so that was very exciting. But Craig and our shared research labs team have already been working through that whole period to put together what is going to be our application for the shared research labs, because just like that story with the Helios Hyperion, that's how we can leverage all of the things that we do in this building, not just for our scientists in stem cell and regenerative medicine as space, but for many investigators across campus in all different kinds of domains. And so thank you, Craig, for leading that initiative so competently. In fact, they did such a great job that the Office of Research just a few weeks ago awarded us a $150,000 seed grant to help get that launched. And again, that was really spearheaded by Craig, and so we're very grateful to his role. So oftentimes, one of the questions that we get, and of course that we try to encourage out of these events, are how can we help from the community, from our audience. So please participate, come to meetings like this. We're going to be holding, we hope, if everything holds with our COVID situation, a couple of open houses later in the year. We are launching a new infusion center that's clinical space that's downstairs here in this building. Also a new CGMP facility that's being built out in Hewitt Hall just across the way. And we hope we're going to have the opportunity to invite people in from the community to be able to tour those and get a sense of that aspect of stem cell and regeneration.
What's a CGMP?
GMP, cell manufacturing facility, which I spoke about at our last event. Thank you, Brian. So please participate, come and see us and see the things that we do. Of course, we are always looking for help in terms of philanthropic donations, so keep us in mind there, because research drives the economy, but not a lot of philanthropic donations go to research, so we're always looking for that. Just a couple of quick words on format then. I was explaining to Craig a moment ago, this is a hybrid lecture, so we have an online audience as well as the people that are in this room tonight. For that reason, even though we generally like the back and forth as we have speakers going, I'm going to ask you to hold your questions until Craig is done. And then we'll do a moderated session at the end, taking questions from our Q&A box for those of you who are online at home, and from those people that are in person in the audience. And so that is going to be our structure. With that, I'd like to more formally introduce Craig, who I shouldn't say this out loud. He's much more important than I am. I always thought he was way senior to me, but we're actually very similar in terms of our timeline. So he received his PhD from UCLA and did time as a postdoc and also a project scientist as UCSD, joining UCI in 2001, which is the same year that I came to UCI. He is currently not only a professor of molecular biology and biochemistry on campus here in the School of Biological Sciences, he's co-director of the Institute for Immunology and a member of the Cancer Research Institute. I've already told you how incredibly important he is to the Stem Cell Research Center, where he helps with our shared research labs and has led all of these initiatives. But he's also the director of the Multiple Sclerosis Research Center. So with that, Craig, welcome.
All right. Well, thank you so much, Eileen. I also want to thank everybody here that's come to listen to my talk, as well as the people online. So again, I want to thank everybody for joining us tonight and just talk a little bit about the immune system, and in particular, how the immune system, particular types of cells of the immune system called T cells, the roles that they play in neurodegenerative diseases, and we'll talk tonight about multiple sclerosis and Alzheimer's disease, and in particular, how we might be able to take advantage of regenerative therapies to alter the impact of these T cells as well as other immune cells in these diseases. So just before I start off, I just want to thank the people in my laboratory. This is a pre-COVID picture, and the lab has changed a bit since then. We haven't had a chance to get a... Sorry, what's that? Oh, it's not showing up on Zoom?
Yeah.
Okay, hold on.
Say stop share and try it again, because it was working.
All right, hold on. Let me see. Let's try that. Does that work now?
Yes.
Okay, sorry about that, guys. Again, not a PhD in technology. Okay, so just want to thank the people in my lab who have been involved in some of the work, and I'll come back to these folks as we go through some of the slides. Okay, so just a quick outline for tonight's presentation. So I'm going to give a very brief introduction to the immune system, including T cells and B cells, part of the adaptive immune system, and then really focus on adaptive immunity in the central nervous system. As we'll talk about, we've known about an important role for other immune cells in the central nervous system, but the role of these adaptive immune cells, particularly in some of these degenerative diseases, is still being investigated. Moreover, we're going to spend some time talking about the influence of a particular stem cell type called human neural stem cells, and their role in provoking the influx of a population of special T cells called regulatory T cells, or Tregs, into the central nervous system for the treatment of multiple sclerosis. We'll switch gears a bit to a different disease, Alzheimer's disease, and talk about the roles of T cells in that disease. And then finally, we'll conclude and talk about some future prospects. So just to get into basic immunology, one of the things I think we all understand, especially after this pandemic, is that our immune system is there to protect us from microbial pathogens, including viruses, fungi, bacteria, and parasites, but it also protects us from tumors. And so we know that there are certain features of tumors that can be recognized by the immune system, and we'll talk a little bit about features of the central nervous system as well that can be recognized by our immune system. And when we think about the sort of mechanisms that are involved in our immune system, we sort of break these down into two classes, one of which we call the innate immune system, and that is a system that recognizes something called pathogen-associated molecular patterns, or PAMPs. And these are sort of features, molecules that are expressed, for example, in bacteria or viruses that basically characterize those cells and give our immune system essentially a hint that there's something wrong, there's an infection taking place, or there's some sort of cell damage that's taking place. These innate immune cells, along with the skin, basically serve as sort of the first line of defense against these sorts of insults. But the system is non-specific, so it recognizes those patterns, but it doesn't give us any sort of memory, doesn't respond to vaccines, things like that. And so, but again, very important, and it includes certain specialized cell types. All of our cells have innate immunity, but there are also specialized cell types, including macrophages, monocytes, dendritic cells, neutrophils, mast cells, et cetera, that all have these kind of innate immune functions that are specialized and brought into particular innate immune contexts. Then we have what we call the adaptive immune system that recognizes what are called antigens. So instead of these molecular patterns that are associated with particular kinds of viruses or bacteria or fungi, instead they recognize something called antigens. And when we talk about antigens, and we'll talk a lot about antigens tonight, that is basically a portmanteau of antibody generators. So antigens initially were discovered in the context of antibodies. So antibodies recognize certain molecules. But the important thing about the adaptive immune system is that it's not recognizing these kind of molecular patterns, it's recognizing these three-dimensional structures. And in fact, if we went to Mars, there was a Mars virus, our immune system would be capable of recognizing these Mars viruses, whereas our innate immune system may not be able to. So it gives us both this unique and incredible specificity, but it also provides memory, and we'll talk a little bit about that as we move forward. So when we think about adaptive immune mechanisms, we can classify those into different forms, one of which is the humoral immune response, and that involves antibodies that bind to soluble antigens. So these are antigens that exist in, for example, the blood or between cells and so forth, and they can then kill or neutralize pathogens, they can kill tumor cells, et cetera. These antibodies are produced by a specialized cell that's called the lymphocyte, in particular B lymphocytes. There's also a cellular adaptive immune response that depends on T cells, and we'll spend a lot of time talking about these cells tonight. And these T cells bind to peptide antigens that are presented on the surface of cells. And this is presented by a molecule called MHC. And MHC presents these little peptides out to these T cells and allows recognition of both of antigens that exist outside of our cells as well as intracellular antigens. And so in a sense, the adaptive immune system also allows us to peer inside of cells. So if a bacteria or a virus wanted to hide out from our immune system, this sort of mechanism allows those pathogens to be recognized even when they're trying to hide out like that. There's also collaboration, and in fact, we know that B cells that produce particularly effective, strong antibodies, they require interactions with T cells, something we call T cell help. And so this is just an example of B cells and B cell development, and I don't want to belabor this point just to say that there is these sort of interactions between B cells and T cells. Notably, those interactions between T cells and B cells within our lymph nodes, that leads to alteration of the B cells' antibodies that it can generate and makes them much stronger. So they bind with much higher affinity. It also changes what we call their isotype. So the function of what those antibodies will do, whether they're involved in protection along mucosal barriers, so for example in the lungs or in the gut, or whether they, basically sort of changes the function of those. And so T cells are important in that sort of collaboration. And this is also very important, these sort of interactions with T cells, very important for the generation of memory B cells. And memory B cells, again, all of us, most of us, I think, have been vaccinated recently, right? So the basis for that is to generate these kinds of memory B cells. We also generate memory T cells as well from those vaccines, and that gives us protective immunity against, in this case, the SARS-CoV-2 virus. So for T cells, and this is just an image showing T cells. As I mentioned, these T cells also recognize antigens like antibodies and B cells do, but instead they're recognizing that as peptides. And these peptides are presented on the surface of a cell. Here we call these a target cell or an antigen-presenting cell in the context of a molecule called MHC, either MHC class 1 or class 2. And the significance of that is that the T cell has on its surface a T cell receptor that when it binds to this peptide antigen presented by MHC, it then gets a signal that causes that cell to become activated. It can undergo what we call clonal expansion, which has nothing to do with Star Wars, but has to do with making a lot of copies of those cells so that you have enough of those cells to be able to respond and clear viral infection. There's two different classes of these cells, one of which are called Cd8 cells, and that's because they have this Cd8 co-receptor on their surface. And these are cells that are generally involved in killing off cells that might be infected with a virus, or they might have some sort of they might be a tumor cell, something like that. And that's another cell type is called the Cd4 cells, and these are what we call helper T cells because, as I showed previously, they're involved in helping B cells to generate antibodies. But these helper T cells also have a lot of other functions. They can express what we call cytokines, and those cytokines are basically released from the cell. They serve as extracellular signals that tell other cells in the immune system how to behave and how to respond appropriately to a particular type of challenge to the immune system. This is just a picture showing, here's a T cell here interacting with, in this case, what we call a dendritic cell, which is part of the innate immune system. And so these kinds of, this interaction then leads to the activation of these T cells so that they can undergo this sort of clonal expansion and generation of these kinds of immune functions. Now, what about the immune system in the central nervous system? So we're going to focus a lot tonight on the CNS. One of the things that's notable about the CNS is that it's considered to be an immunologically privileged site in which our peripheral immune cells, like T cells and B cells, are really not supposed to have access. And the thinking, I guess, behind that is that neurons, for example, don't regenerate very efficiently. Doesn't make a lot of sense to kill those cells off like you might say cells in the lumen of the gut or something like that. So there is this concept that there's immunological privilege, and in fact, we know that there's something called a blood-brain barrier, or the BBB, which is basically a series of tight junctions that exist in cells in our blood vessels that move throughout the brain. And the result of those tight junctions is that you don't get easy access. You don't get large molecules like antibodies. You don't get cells like T cells and B cells entering into the brain very readily, unless there's some kind of viral infection or something going on, and then the blood-brain barrier can break down. Moreover, that we know that the CNS has basically these native innate immune cells, and particularly we know about microglia and astrocytes, and they play important roles in a number of different features of brain development and protection. But more recently, there's evidence that suggests that there are native populations of B cells that exist within the brain, particularly in this area of the dura, within the meningeal membranes of the CNS. Work from Jonathan Kipnis and Marco Colonna's lab, and more recent work as well, showing that there may be some of these cells, these immune cells, that are developing within the bone marrow of the skull. Moreover, we know that T cells can gain access to the CNS via the choroid plexus, which is a site where cerebrospinal fluid is generated. So we know that there is this sort of immune privilege, but also that there is this sort of regular interactions between cells of the immune system, including from the peripheral immune system into the CNS, as well as this more recent data that suggests that there are actually B cells that exist natively within the CNS. So one of the questions I think we all ask ourselves is, what are the physiological and pathological consequences of such immune cells that exist within the CNS or have entry into the CNS? And one example of that is a disease called multiple sclerosis, which we've known for a long time involves recruitment of these immune cells from the periphery into the CNS. MS is a chronic inflammatory autoimmune disease of the CNS. And so when we talk about autoimmune disease, what we mean is that our T cells and our B cells and our antibodies basically start to recognize our own tissues, what they really shouldn't, right? They should be specific for viruses or tumors or something like that. But it turns out that there are some mistakes that are made, and that can result in these kinds of autoimmune diseases, including multiple sclerosis. And the result of this is that you get this infiltration of immune cells that lead to the destruction of myelin, which surrounds neurons, and that results in defective neurological function. What I mean by that is here's just showing what a normal neuron looks like with its axons, and those axons have this essentially insulation. If you think about an electrical wire, it has this plastic insulation around the outside so that it doesn't conduct, it doesn't short out and start to cause a fire or something like that. Well, our neurons have similar kinds of insulation with this myelin, and that myelin is produced by a special cell type that are called oligodendrocytes. When we have folks that have MS, they've got T cells that get recruited into the central nervous system that recognize myelin inappropriately. They start to become activated against that myelin, and they start to activate, for example, microglia and astrocytes. Ultimately, that leads to the loss of this myelin, and as a result, nerve conduction doesn't work very effectively. And so people that have MS can develop paralysis as a result of that, and it can be ultimately lethal. Most of the folks that develop MS are treated with drugs that block T cell activation or T cell recruitment, either out of lymph nodes or into the CNS. And that's relatively effective, but it's not a cure. And oftentimes people will, the drugs will lose effectiveness in certain patients. And so that's certainly one problem. In addition, there are different forms of MS, oftentimes where you may start off with one form of MS that's treatable with these kinds of drugs, but over time, there's so much damage that's caused by this unrelenting inflammation within the central nervous system that those drugs no longer are effective, and there's just so much damage that those folks really, there aren't very many options for them. And so that's where we get interested in potentially using stem cells for treatment of these folks. So one question that I think we have to think about is, what is it that causes MS? And so this is a question that's been addressed by, for decades by numbers of researchers all over the world. But things that we know about this, one is that there are T cells that are reactive against myelin-derived antigens that enter into the brain and into the spinal cord, and they basically start to attack the cells that are producing myelin, these oligodendrocytes. And this basically requires a breakdown in what we call immunological tolerance. Our body is not supposed to attack our own tissues. Our immune system is supposed to be tolerant of that, and somehow that tolerance is broken. And importantly, as I was mentioning, in these sort of later stages of MS, these chronic and progressive forms of MS, this inflammation over time leads to this destruction not only of the myelin, but it also can lead to loss of neurons. And that can really cause problems. So again, that sort of led our lab and many other labs to look at possibly applying stem cells to be able to treat some of these folks, particularly in some of these chronic and progressive forms of the disease. So how does this work? What's the mechanism here? So here we start off in the thymus, which is where our T cells develop. Early on, the thymus is a gland that exists right behind our heart, and these T cells start to develop even as we're in utero. And importantly, the T cells, if they are overtly autoreactive, so if they can be activated by self-antigens, they usually die off, okay? And that's sort of an important tolerance mechanism that prevents autoreactivity. But on top of that, you can also get generation of what are called regulatory T cells. And we'll spend a lot of time in this part of the talk talking about these Tregs. And the Tregs, unlike these what we call conventional T cells, they're anti-inflammatory, and their role is really to block other T cells and other immune cells from causing autoreactivity. Okay, so these T cells, once they're educated in the thymus, they then go into the periphery, into a lymph node, into the blood. They basically move around from lymph node to lymph node in the blood and the lymphatic system, trying to find the antigen that they can recognize. And each of these T cells, just like with antibodies, each of those T cells, is very, very specific for a particular kind of antigen. Once they recognize that antigen, they'll undergo this sort of clonal expansion, which we'll talk about. But the idea here is that we have these T cells. They should be tolerant in the periphery, but not becoming activated unless they have their T cell receptor here on the surface of this T cell interacting with peptide MHC, as we talked about. I'll get around that. They have these sort of cytokines that I talked about. And the result of that is that they start to undergo clonal expansion, make lots of copies of themselves. And in this case, we have what are called helper type 1 and helper type 17 cells that are pro-inflammatory and are involved in normally in antiviral, antibacterial kinds of responses, as well as these Tregs that can restrict these if these conventional T cells are autoreactive. Now in the case of MS, some of these T cells that are autoreactive will traverse their way across the blood-brain barrier, enter into the central nervous system, and then the result of that is that they start to activate cells within the CNS, including microglia that leads, and astrocytes that leads to the demyelination or the stripping of this myelin along the axons of these neurons, and that ultimately can lead to this kind of loss of function. Tregs are important, we know, particularly in MS patients, because they're there to block the activity of these T cells. And there's still a lot of interest in understanding how we can take advantage of these Tregs to try to treat folks and try to reestablish tolerance against some of these myelin antigens to try to block this sort of ongoing damage that's happening within the CNS. So one of the things that we've been focused on for some time is looking at a special type of stem cell called neural stem cells. And we're looking at those as a potential therapeutic for treating neurological disorders, in particular looking at this disease, multiple sclerosis. And these cells, like many stem cells, are self-renewing. And neural stem cells in particular have the capacity to give rise to different types of cells within the CNS, including neurons, astrocytes, and these oligodendrocytes. And in particular, as I mentioned, these oligodendrocytes are important for myelination. And work that was done in Tom Lane's lab early on, he was able to show that using what we call Cingenic mouse neural stem cells, so these are basically derived from a mouse that's genetically identical to the mouse in which we're going to transplant these cells into, and using a mouse model of MS in which virus is put into the brain, resulting in a bunch of T cells going into the brain to try to fight off the virus, but in so doing, there's a lot of collateral damage that leads to demyelination, looks something like MS. And so Tom's lab had found that if you transplant these Cingenic mouse neural stem cells into the CNS of these mice that are undergoing this virus-induced demyelination, that would restore function, that would basically rescue those mice. That was also work that was additionally shown by Laura McIntyre and Julio Ayala Angulo from my laboratory later on. But then sort of an interesting experiment that was done also in collaboration with Tom's lab was to take human neural precursor cells, neural stem cells, and to, in this case, they're what we would call xenogenic, meaning that they're, not only are they different, in terms of, their genetics, but they're from a completely different species as well. And so the consideration there is that you're going to have immune rejection, right? The immune system of the mouse is going to recognize these foreign cells, going to reject those, and we're not going to get any response. But in fact, what Tom's lab was able to show is that in this, again, this viral model of MS, that transplanted these xenogenic neural stem cells led to remyelination, and that was associated with the generation of a large number of regulatory T cells that, moreover, that if we got rid of these Tregs, the effect of those neural precursor cells went away. So neural stem cells were important, but in this case, with this xenotransplant, it wasn't so much what we would call cell replacement, where those stem cells are differentiating into oligodendrocytes, and those are then inducing remyelination. But instead, there was this immunomodulatory effect that involved these regulatory T cells. So we also wanted to characterize this in a context of a different model of MS that's something called experimental autoimmune encephalomyelitis, or EAE, where you basically take mice and you immunize them with myelin antigens. The immune system recognizes those, you get T cells that go into the brain, and those T cells start to attack the myelin. And in this case here, we're staining these mouse brain sections with a stain called Luxol Fast Blue that stains the myelin. And here in control mice, you can see that there's these areas that are white. So these are basically demyelinated, lots of loss of myelin. But if those mice had received human neural stem cells 3 weeks prior, the amount of this sort of demyelination is considerably less. And so you can see a lot more of this sort of blue staining myelin. And this is work that was done by the lab, including Quan Pham and Estelle Vu. And this is just showing here that this seems to be sort of focal, so that the injection site where we actually inject these into the spinal cords, these neural stem cells, where we inject them, we seem to see the highest level of response. But there is a response all over, and I'll show in a couple of images later that we do see that there are regulatory T cells that get recruited throughout the spinal cord in these mice. And just to show that there are Tregs in this case, here we're looking at the total number of Cd4 cells. So again, Cd4 cells generally are bad in MS because we don't want T cells to go into our brain and start to attack these cells that are producing myelin. When these mice get these NSCs, the overall number of these Cd4 T cells is significantly reduced. On the other hand, if we look at the number of Tregs, there's a statistically significant increase in the number of these Tregs that get recruited into the cervical lymph nodes, which are basically lymph nodes that are draining from the central nervous system, from the spinal cords of these mice. So that result basically tells us that again, as with the viral model, that these Tregs are important and they are induced by these neural stem cells. So Laura, a former graduate student in my laboratory, asked, I think, a really important question, which is, number one, what are these neural stem cells doing to induce these Tregs to go into the central nervous system? And then a second question that we haven't really been able to address so much yet is, what are those Tregs actually doing once they get into the central nervous system? But to address the first question, what are the NSCs doing to bring these Tregs in? She decided to set up a co-culture. So basically an ex vivo system where she could take neural stem cells, mix them with a source of immune cells, in this case from the spleen, where we've got T cells and B cells and innate immune cells like dendritic cells, mixing those with these neural stem cells, and then seeing what happens over time. Does that have any change in the population of T cells that you get? And here we're looking at different markers for T cells, and in particular we're focusing on something called FOXP3 as well as CD25. These are both proteins that are expressed within these regulatory T cells. And the important point here was that this gave us a way to look to see, do these NSCs provoke or in some way induce this population of Tregs? And so this is just showing that as you increase the amount of these NSCs relative to these splenocytes, you see that there is also an increase in the number of these regulatory T cells. So we can recapitulate what's happening within the CNS of these mice in these ex vivo co-culture system. And importantly, in work by Scott Grelak and Brian Wee from my lab, we looked to see whether or not those NSCs needed to be viable. Because one of the questions is, well, what's the function? How are these NSCs actually causing these T cells to become, recruited into the CNS and to become these Tregs? And so one of the thoughts was, well, maybe they're actively producing cytokines, things like that are causing these. But we decided to do an experiment just to say, what do the cells even need to be viable? And even if they're not viable, we were still able to get induction of these Tregs, which suggested that there might be something going on. Perhaps it might be at the level of peptide antigens. And so that made us think we might want to look to see whether these NSCs might serve as a source of these myelin antigens. And so one of the ways that we could address this question about myelin antigens being and neural antigens being expressed within these NSCs was to think a little bit about something called the clonal selection theory, right? So this is something that was first proposed by Frank McFarland Burnett and David Talmage. And the idea is that if you have a particular T cell or B cell, there may be many different what we call clones, meaning that each one of those has a particular type of antigen that it will recognize, and such that if we have billions of these different T cells or B cells and they all have their own specificities, they're not going to be particularly useful in protecting us from, say, a viral infection. But if Burnett's idea was if they recognize one of these antigens, what they'll do is they'll start to undergo cell division and make many, many copies of themselves until there are enough of those cells to be effective and to be able to control a virus. And so our question was, is this something that's happening in response to these neural stem cells? And to address that question, and this is just showing an example of here where this particular, let's say it's a B cell, it recognizes antigen X. Once it does so, it undergoes a lot of cell division and makes lots and lots of copies of itself, lots of clones. And since we can actually look at that specificity based on the particular antibody molecule, or in a T cell, we can look at something called the T cell receptor and look specifically at the molecule itself, we can determine whether there's any sort of change in the populations of clones that exist in these co-cultures. And so that was something that was done here by Laura in collaboration with Keen Nguyen from Kai Kessenbrock's lab. Basically sequenced the TCR beta chains of all of these different T cells to see is there any similarities or differences in the repertoire of different T cell receptors in those populations of T cells. And what she found was that if you looked at sort of a general population of Tregs that are produced in response to certain culture conditions, there are a number of those receptors that are in common with those that we see in these NSC-induced Tregs, but there are also some that are unique to these induced Tregs versus these a whole large population of T cell receptor sequences that are specific for these NSC Tregs. So that this result basically tells us that there appears to be a difference in the antigenic specificity, or what these T cells recognize when we do this sort of co-culture. Again, sort of supporting the idea that what these NSCs do is to supply antigen. And to really address that question, we used a sort of a genetic, immunological trick where we can take the T cell receptor in mice and we can restrict it to a single specificity using T-cell receptor transgenic mice. So these are C57 black 6 mice, so they're basically wild-type mice. And if we take those, as I showed before, when we do this co-culture, we get this expansion of these Tregs. If you do the same thing where you have a T-cell receptor that is specific for chicken ovalbumin, so obviously not anything that has to do with the brain, you don't get this expansion of these Tregs. However, if you do so with a mouse that has a receptor, a T-cell receptor, and only recognizes MOG, it is cross-reactive with neurofilament, so these are both CNS antigens. You do see the expansion. It's not as efficient as you see with a polyclonal, TCR repertoire, meaning that, there's probably some more expansion that has to take place. But the important point of this is that this definitely proves the point that antigen specificity is something that's being provided by these neural stem cells. And moreover, and just I don't want to get into sort of the weeds on this, but just an additional sort of experiment is what's the origin of these regulatory T cells? What population do they come from? And Scott did some experiments where he basically got rid of all of the pre-existing Tregs just to see whether or not co-culture with these NSCs could induce Tregs. And in fact, even if you get rid of those pre-existing Tregs, we do see this expansion, suggesting that those cells can expand on their own. We also think that they might also expand from what are called X-Tregs. But again, I don't want to get into the weeds on that. All right, so just kind of looking at this question about, so we know that these NSCs are involved in expansion of these Tregs. What are those Tregs doing once they get into the central nervous system? Do they have some kind of are they just being anti-inflammatory, as we know that they can do? They can block these autoreactive T cells, or do they do more? And there's evidence from several laboratories that suggests that regulatory T cells are not just anti-inflammatory, but they can also induce repair. They can secrete certain molecules that will induce the repair pathways to take place. And this is work that was done in collaboration with Shiva Othi from Michael Cahalan's lab here on campus. And Mike's lab is years ago developed this technology called intravital two-photon microscopy, where we can look at interactions between immune cells in live mice. And so Shiva took advantage of that technology using the spinal cord explants to really look at immune cell interactions within the spinal cords of these mice undergoing this MS-like disease. And so this is an experiment just looking at mice where we've got these Tregs are labeled with something called FOXP3-GFP. So this is a fluorescent protein that's expressed specifically in these Tregs. And looking at, as well, these human neural stem cells that are labeled in red. And so this is basically an image across the spinal cords of mice that have gotten these. And just stitching hundreds of different images, but you can see that there's a pretty broad population of these Tregs that get recruited into the spinal cord in response to these neural stem cell transplants. And then the question was, what are they actually doing when they get there? And so this is an experiment that I think is kind of cool. Here we have mice that are non-transplanted, and again, in green we've got these Tregs. In this case, in red, we have cells that are labeled with PLP TD tomato, so it's basically oligodendrocytes or oligodendrocyte precursor cells. And so on the left, these are control mice that were not transplanted with these human neural stem cells, and you can see there's plenty of Tregs there, but they're fairly distant from these